Hello everyone, welcome to the NPTEL online certification course on fundamentals of food process engineering. Since the last two classes we were discussing about the non-thermal processing, which are the processing methods by which without heat treatment we will try to inactivate the microorganism, and so far we have discussed about high pressure processing and pulse electric field. So, we have seen that how in these two method we can inactivate the microorganism and what are the process parameters that actually can be varied during different inactivation of microorganism, because not all microorganism need the same intensity of application of any particular process, because it varies from types of microorganism. So, all this we have discussed, and today we will see some more techniques, non-thermal techniques, by which the inactivation of microorganism can be done. So, this technique high pressure processing and pulse electric field technology, this we have discussed. Pulse light we will discuss today, followed by irradiation, ozone and cold plasma technology. And at the end we will discuss what is hurdle technology. So, first we will discuss the pulse light technology. What is pulse light? Pulse light actually a broad spectrum white light, and this has capability of sterilizing the material. Basically this works on the surface of packaging material or the food contact surfaces. That means the equipments or utensils that we use for processing the food, or for example the the conveyor where the food or fresh vegetables, or after processing we want to convey that from one section to the other section. So, for sterilizing all those equipment and for sterilizing the packaging material, we use the pulse light technology. Traditionally in the industry, this sterilization of packaging material is being done by hydrogen peroxide, but we know that hydrogen peroxide leaves the residue both in the packaging material or in the food itself, and that is harmful. That is why pulse light technology is becoming important nowadays, and it is very effective also. We can see that pulse light is can be considered from 200 nanometer to almost 1200 or 1250 nanometer. This is the range, and it can include the UV from 200 to 400, then 400 to 700 the visible light spectrum and the infrared spectrum. Although the inactivation of microorganism by pulse light technology fully not understood, the technology or how they actually have the detrimental effect on the microorganism is not fully understood by now, but whatever may be the case, UV definitely playing a significant role, right. So, that is understood that UV is definitely playing a significant role in decreasing the microbial contamination on the surface using the pulse light treatment. So, we will see now what are the parameters associated with it. So, light pulses application is a non-thermal method of food preservation, involve the use of intense and short duration pulses. These are very intense pulse and short duration pulses of broad spectrum white light. We have seen the range, it starts from 200 nanometer to almost 1250 nanometer. Light pulses are used to sterilize the microbial population on the surface of the packaging material and also on the surface of the processing equipment and food. Light pulses reduces or eliminate the need of chemical disinfectants and preservative, because this is the conventional method. We use that disinfectants and preservative, and those obviously have some chemical residue. So, that we can eliminate by this use, as I mentioned about H2O2. And inactivation of selected resistant microorganism requires treatment with the complete spectrum, that means the range that we are having 200 to 1250, and other microorganisms are inactivated with filtered spectrum. That means from this broad range, that is 200 to 1250, not all the spectrum is needed for kill all the microbes. It is specific to some microbes. So, some selected or filtered spectrum is needed, and in some cases the whole complete spectrum is required. And the duration of pulses ranges from 1 microsecond to 0.1 microsecond. So, the duration of applying pulses is also very less. The flushes are typically applied at a rate of 1 to 20 flushes per second, and at times one pulse is enough to inactivate the microorganism on the surface. So, we can say that this is an effective method, and cost involvement is also very less, okay, and there is no residue. So, removal of the residue or chemical preservative cost, that is also can be nullified. So, that way this is very helpful method in the industry. Now, the pulse slide system consists of several components. So, what are those components? One is the high voltage power supply that provide the electrical power to the storage capacitor. There is a storage capacitor to store the electrical energy for the flush lamp. We generally use an inert gas lamp to have that intense light pulses, and that lamp is actually gradually glow from the low intensity to the high intensity, and the pulse forming network is there. This pulse forming network determines the pulse shape, that of which type or which pattern of the shape is generated, and the spectrum characteristics, that means the spectrum of what length we are using, okay, where it is the total pulse we are using, total spectrum we are using, or a filtered spectrum we are using, and the pulse shape. There is a gas discharge flash lamp is there, that is the inert gas lamp, and a trigger signal is there. So, this initiate the discharging of the electrical energy to the flash lamp, which is the key element of the pulse light unit. Now, when we have a lamp of the inert gas and we initiate the discharge of the electrical energy, then that electrons will interact with the atoms of the gas and they produce the high intensity white broad spectrum light that helps in the inactivation of microorganism, and that generate for a very small duration, as we mentioned about the millisecond to the microsecond range. So, this is the system. Here we have the high voltage DC power supply. We have a capacitor, energy storage capacitor, that stores the energy, and when we apply the switch, so the flushed lamp here, it starts generating the intensity of the light from the low to high, and a flash will be generated. So, that radiant energy coming to the target of the food contact material, any surface or the packaging material, etc. And there is a pulse shaping inductor which indicate or which determines that what kind of pulse shape will be generated. Okay. So, this is the functional diagram of pulse light treatment. So, there are certain terms that we need to know before applying this pulse light treatment to any contact, food contact surfaces or of packaging material, because on this parameter it depends, the intensity of the action of the pulse light will depend. So, one is the fluence rate. Fluence rate means the energy received from the lamp by the sample per unit area per second. Okay. So, that means per unit area per unit time, that is energy received from the lamp, that is called the fluence rate. Now, fluence per dose signifies energy received from the lamp by the sample per unit area during the treatment. Okay. So, this is considering the whole treatment, what is coming, not per unit time. Then there is pulse width. Pulse width is time interval, that is fraction of second, during which the energy is delivered, that is the pulse width. Exposure time, that is time period in second during which the treatment is given. Okay. So, time interval, that is the fraction of second during which the energy is delivered, and the time period in second during which the treatment is given, this is the exposure time. What is peak power? It is measured at the pulse energy divided by the pulse duration, because it generates a peak and then it gradually diminishes. So, we have to determine the peak power by the pulse energy divided by the pulse duration. Then pulse repetition rate. So, number of pulses per second, okay, that is the frequency of the pulses in hertz and commonly expressed as pulse per second. So, this is the pulse repetition rate, that is in per second how many pulses are generated. So, these are the technical terms that we need to look for while designing the pulse light system. Now, the treatment of products by using short duration intense pulse light is based on the penetration of the light through the product. Okay. So, as we are mentioning that basically it acts on the surfaces. So, it can, if it can penetrate a little, so what will be that depth and how it can work there. So, light penetrates the surface of material according to this law. Okay. So, it says that I equal to 1 minus r into I 0 e to the power minus x. I is the energy intensity of the light transmitted to a distance below the surface. Okay. So, if this is a surface and we are applying this intense radiation. So, what will be the in this direction, what is coming in. So, the energy intensity of the light transmitted to a distance below the surface. R is the surface coefficient of reflection. So, because what is coming in, some will be reflected, some will be transmitted. So, the R is the surface coefficient of reflection. I 0 is the intensity of incident upon the surface. So, I 0 which is coming, intensity of the incident radiation. Here it is light, intensity of the light which is coming, okay, and x is the extinction coefficient which determines the opacity of the material. So, opacity of the material defined by x, that means that define that how much the light can penetrate. So, if we see that we have that diagram. Okay. So, first we will see that and then we will go back to those equation. So, incident radiation when it is coming, then it is reflected some part and some part is transmitted. So, it transmitted at a distance is x, that we have defined their extinction coefficient, and there is d. So, here it is the absorbed energy, okay, and this is the transmitted energy E x. So, this is how when the pulse light comes in on the surface of a material. So, the reflection, transmission and absorption of the light radiation takes place. Now, we will go back to, so this expression we have discussed. So, the next is the heat energy dissipated per unit area E d. The heat energy dissipated per unit area will be E d, that is equal to 1 minus R into I 0, the intensity of incident light, into 1 minus e to the power minus d. So, energy dissipated per unit area E d, or that energy absorbed at a depth d, is described by E d equal to this 1 minus R I 0 1 minus e to the power minus d, and heat transferred by conduction E x is described by E x equal to A, that is the area of the exposure, into K, that is thermal conductivity of the material, and T, which is time, into dt by dx. So, dt, this dt is the temperature difference between these the in and out of that layer. So, the distance it crosses, and dx is the heat transfer distance. So, across the distance dx, if there is dt temperature gradient, so the heat transferred by conduction will be this much, k a t dt by dx. Now, we have described already that what will be the energy, incident energy, then reflected energy, transmitted energy or conducted, and then absorbed or dissipated at the distance d. Okay. So, then there is factors affecting the inactivation of microorganism. Now, this depends on three things. First is type of microorganism, then the interaction between light and substrate, and the distance from the light source. So, type of microorganism, because every microorganism, or if we consider their characteristics is different, some have, suppose cell wall membrane is strong, some does not have cell wall. So, there are many kind of microbes, and what is actually defined, that what type of the microbes is sensitive to particular spectrum of the light. So, that is why we have mentioned initially that to inactivate some microorganism we need a whole spectrum of the light, and for some we need the filtered spectrum. So, that specifically depends on what kind of microorganism is present. Then interaction between the light and substrate, it actually based on whether the material is transparent, whether the material is opaque, whether their surface is rough, or it is having any kind of interaction with the light, it is penetrating at the surface or not. So, so many parameter, it depends on the substrate, that is what we are taking. And finally, the distance from the light source. So, the distance from the light source is again important, so that it can have its energy and its effect on the surface to a higher extent or a lower extent. So, we have some application related to pulse light in the food industry. So, it kills the bacteria and spore, as we have mentioned initially that the type of microorganism is very important. So, now we will see that what type of microorganism on which the pulse light is very effective. So, it kills the bacteria, okay, and spores, while UV causes damage to the nucleic acid and other components of the cell. The instantaneous heating of the cell results in the rupture of the cell wall or lysing. So, we have mentioned initially that whether the effect of the microbial destruction is because of UV only or any other mechanism, that is not very clearly understood, but this has been assumed that the initial effect that causes damage to the nucleic acid and in the cell wall destruction, that causes because of the UV spectrum, and this causes damage to the nucleic acid, but because of this energy absorption there will be high amount of heat generation as well. So, that heat causes the results in the rupture of the cell wall, and eventually UV attack the nucleic acid and the destruction happens. So, this is a combined effect of the UV and the heat and maybe some other parameter which is not very clearly known as of now, because there are effect of other spectrum as well. Now, for material is concerned, we can see that transparent and colored food material, if it is transparent material and colored food material, so refraction is particularly relevant, because we have the incident and then transmittance and then reflection. So, if for the transparent and colored material refraction is relevant, opaque food material reflection will be the concern. So, we need to see that how much will be actually transmitted. For smooth surface specular reflection will be there. For rough surfaces diffuse reflection will be there when the surface is rough, and for translucent material scattering will be there. Okay. So, therefore, from this we can see that all such cases transmittance will be varied, and based on transmittance and absorption the destruction of the cell will be there. So, we need to analyze properly that what is the interaction of the light, pulse light and the surface. So, therefore, this pulse light can be used in disinfection of air and water for aseptic application. The thing we previously done by some chemical disinfectant. So, that is now used by pulse light treatment. Inactivation of spoilage and pathogenic microorganism in fluid food, this is being done. Fluid food, because in that case we can destroy the surface microorganism. For a solid we have to penetrate deep, and that may not be done by the pulse light treatment. That is why we consider on the microorganism, pathogenic microorganism of the fluid food. Then surface microbial control. So, disinfection of the utensils and surface, as we mentioned, disinfection of packaging and closure, surface decontamination of the fruits and vegetables. So, suppose we want to convey this from one location to the other, and while conveying it is exposed to the pulse light treatment, so that the surface decontamination can be takes place, those things can be done. And also pulse light is used in the Huddle treatment. So, Huddle treatment at the end of this non-thermal we will discuss in a small way. So, there we will elaborate what is Huddle treatment is. So, this is the use of the pulse light in food industry. So, we will stop here and we will move on in the next lecture with one more topic of non-thermal preservation. Thank you.